Howdy, my name is Noah Holkeboer. I'm a third-year student here at Grand Valley. I'm double majoring in cell and molecular biology and biology with an emphasis in ecology and evolution. I'm also a student in the Honors College. Today I'm going to be walking you through the research that I've been doing for the past 3 years. A little bit different for Scholars Day. Instead of focusing on one project, I'm going to kind of take you from the start of my research to what I'm working on right now. And the name of my presentation is Seeing the Forest for the A's, T's, C's, and G's, Investigating Tree Architecture at Multiple Levels of Biological Organization. And up front, I want to thank my research mentor, Dr. Gary Greer. So how did it all start? How did I even get involved in this? Well, basically, I just asked. My freshman year in my honors sequence, first semester, Professor Greer was guest lecturing in my big history class. And at the end of his guest lecture, he chatted a little bit about the research he was doing, and I thought it sounded really interesting. And so as a nervous kind of freshman, I went up after and asked him if he knew of any other professors that were looking for people to do research, or if maybe there was any room on maybe a future project of his. And he just said, well, we have a research meeting this Friday, you should be there. And I showed up, and I've been falling in love with tree research ever since. I got pretty lucky in that the project I joined was kind of still in its infancy, and so I got to sit at the conference room table and figure out experimental design, got to pick what species we were going to look at. I got to have a voice in what the actual project I was going to be working on was, which was amazing. I didn't just kind of get to do the technical skills of the research. I actually got to kind of participate in the scientific method part of planning it all out. Before I dive into that project, I want to just give you a little general background on the topic first. So first off, the conductive cells, that is xylem, of woody plants have two main functions. It's got to transport water, and it's got to provide structural support. And these two factors, optimization for them can be assessed by examining branching points. So if you look at this image here, you can see that we have a branching point, and before it is what we call the mother, and the two branches afterwards are called the daughters. And we call a branching point like this a node. Previously, all this research on this has either been done destructively by cutting the tree down or taking a limb off, or very expensively, somewhat recently, through high-end 3D imaging stuff that often takes a lot of equipment, skilled operators, and is just costly. So, in theory, a simple photo-based method that is non-destructive and just uses a normal camera to just take pictures of branching points could allow for a lot of new avenues of inquiry, such as longitudinal studies, or easier longitudinal studies, because if you're not cutting a tree down, you can come visit it over time and see how it changes, or just gathering a lot of data fairly quickly, whether that's examining a whole plot or trying to get some citizen science involved, or just easy work for undergraduates. A simple method would be very useful. So this exploratory study that I was a part of, the goal was simply to test if photo-based methods would provide any sort of useful data, specifically to see if we could distinguish species-specific optimizations for those tasks, hydraulic efficiency and structural support. Basically, could we take pictures of trees and tell if different species branched differently? The methods were fairly simple. First, we took photos of the first node, the first branching point of 4 species. We looked at Ficus grandifolia, American beech, Quercus rubra, red oak, Acer sacrum, sugar maple, and Ginkgo biloba, ginkgo or the maidenhair tree. Now, the first 3 of those are native, but ginkgo is a little bit of an interesting outgroup. It's kind of an ancient gymnosperm species. It isn't native, it's only planted kind of in cities, but we kept it as an interesting outgroup. Second, those photos that we took were measured in a program called ImageJ. If you look there on the left, you can see the mother and daughter, this, like I talked about earlier, and also the angle between those daughters, what we call the node angle. And so we measured that in ImageJ, which is pretty simple. It's basically click and drag, and after you scale it, it'll give you a measurement in whatever unit you scaled it in. And then third and finally, we just did data analysis for 3 models. Basically, to actually get the hydraulic efficiency or structural support data or optimization data, you need to actually run those simple measurements through various models. So first we use a model called Murray's Law, which is fairly simple, but it is really examining for hydraulic efficiency. Then we have the uniform stress model, which takes into account branch angle and is a measure of structural support, specifically, or it's a measure of optimization for structural support, specifically the support of limbs branching out to the side. And third, we have the West-Brown-Enquist model, which is about structural support again, but comes at it from a mass perspective. Specifically, can the mother below the branching point support the mass of everything above the branching point? So this is kind of what our results look like for this study, and I'm only going to do this. I'm going to cover this briefly, not get too lost in the weeds with all the numbers and statistics. But first off, I just want to point out that the regressions for all of this were very significant, had very high adjusted R-squares, above 0.9, and had significant p-values, which in itself speaks about some of the utility, or at least the validity of photographs. And then we use something called an analysis of covariance, an ANCOVA, to compare different slopes. But let's look at this figure right here and just break this one down. So on the x-axis, we have the mothers have been run through the model, and on the y-axis, we have the daughters being run through the model. In this case, we're looking at the Murray's Law, that's the hydraulic efficiency model, and we are looking at it for beach. In simple terms, the red line that you see with the triangles represents optimization for that model. It represents being fully hydraulically efficient. The blue line represents what we actually observed in beach. Now, beach is not at that red line, it's slightly under it. Now, that's exhibiting what we call a safety margin. Basically, being on that red line, being optimized, fully optimized for hydraulic efficiency. That's great and all, but it also comes with some risks. Being fully optimized carries with it a risk, more risk of suffering from drought. That if a drought comes through, it'll do more damage to the tree. And so beach, in this case, is keeping a safety margin. It's slightly under-optimized. Now, zooming out a little bit more, this figure right here shows all the results for all four species and all three models. So, like I said with beach just now, that first cell for Murray's Law, negative allometry is the more technical term for this under-optimization or this safety margin that I was talking about. So if we look for beach, all three models have this safety margin, and it's the same story for red oak. So we see that for Murray's Law, West-Brown-Enquist, and uniform stress model, all three are keeping this safety margin. So beech is keeping a safety margin for drought resistance. It's keeping a safety margin so that its branches are less likely to break off under their own weight. But we look at sugar maple, and the story is different. See, in all three cases, sugar maple is at isometry, and that's what we call being without a safety margin. So in that case, the red line, the blue line for Acer sacrum, sugar maple, would be right on it. It's optimized. It's fully optimized, and so it has no safety margin. So it's at more risk of taking damage if a drought comes through, and its branches are theoretically at a greater risk of breaking under their own weight. And then looking at ginkgo biloba, here we have a mix. At least for Murray's Law and the West-Brown-Enquist, we see marginally significant safety margin being kept. And so that's an interesting thing to note, that we're seeing a difference between the models. What does this all mean? Well, first off, there's a lot of evidence here to say photographs can provide some sort of useful data. They can detect species-specific optimizations. We could see that beech and oak were optimizing differently than sugar maple and ginkgo were. And perhaps we might be able to detect these intra-species differences between the models. You know, looking at ginkgo, it seems like we could tell that it optimized differently for some models versus others. And so at the end of the day, this is showing us that these photos can tell us something about these trees. And then the next logical step might be to kind of validate that further, but COVID didn't necessarily care about the next logical step, and it interrupted a lot. So GVSU fully closed the day before the first in-person real conference I was going to present at, so that got delayed and moved virtually. You can see a picture there of me filming from my room at my parents' house, a conference. Which is a bummer, but I'm glad I still got to participate. And also, I had summer work planned, and I had to pivot and figure out a project that I could do fully social distanced and solo. And so what better way to be social distanced than to be surrounded by no one but trees? Enter a forest plot. So what we decided to do that summer was kind of do a proof of concept of one of the benefits of a photographic method. One of the benefits is we can get a lot of data quickly. And so I established a forest plot in the GVSU ravines. You can see in the map there, it's just a little bit deeper than the Arboretum. And on the right there, you can see a really nice picture of me in my plot taking pictures of the trees. So the methods for it were fairly simple. Basically, I established plot boundaries, and then I mapped and took the diameter or breast height of all the trees, and then took photographs of the first branching point, similar to how I did for the exploratory study. Measured the photos in image J in the same way, and then analyzed the data in a very similar way to the experimental study. There were a couple different things that we looked at, but I'll kind of hone in on the key results here. Basically, the two dominant species in the plot optimize fairly differently from each other. We look at Acer sacrum. It's optimizing much more for the support of its limbs. And you can see that in this figure because that green line is Acer sacrum, and it is kind of pulled up towards the uniform stress model, which you can see on the y-axis. And then Quercus rubra, red oak, you can see that red line, which is red oak, is much closer to the x-axis, which is Murray's law, or Murray's ratio, and that's a measure of hydraulic efficiency. So the key takeaway here is that the two most dominant species in this plot, they are falling into different niches when it comes to their branching architecture. And why that might be is not fully clear. Perhaps sugar maple can afford to be optimized one way because it's being sheltered by red oak, or maybe vice versa. That's not fully clear at this point. That's more just hypotheses, but it is very interesting that the two dominant species in this plot took different paths. And this is just mostly further support of the fact that photographs can provide useful data. Here we, again, in a completely different setting with different trees, we see species optimizing differently from each other, and simple photos being able to detect that. Now, there are also some next steps we want to take in the plot. We're actually not done with the work we want to do there. We want to map it in much more detail. I made a pen and paper map when I was out there working in it, but we want to use kind of some GIS techniques to map it out in detail. And that's been Peter Wampler has been helping us out with that, and it's a work in progress, but I'm really excited about it. The idea is that by having a kind of a scientifically detailed map, not just one that will help you find the trees in the future, we could then start running some analyses looking at whether trees that are crowded, that is, they have a lot of other trees right next to them, branch differently than trees that are out in the open. They're kind of far away from the other trees. We could look at if species are kind of clumping together and how that might play into their branching. And perhaps we could even pull down some data from satellite imagery or the slope of the ravines and see if trees optimize differently based on their kind of micro geographic conditions. So that's really exciting. And then probably the most important step that we've taken in terms of validating a photographic method are destructive conformations. And I'm going to talk about one destructive conformation that we've done. And the goal here was simply to test photo measurements against direct measurements. And so we did that, or it was done by cutting down a red maple tree and cutting the node sections out, taking pictures of them, and also measuring them directly and comparing those measurements. Now, I wasn't involved in cutting this tree down, but I did measure the nodes and measure the photos. And when we did this, we found a strong correlation between the photo measurements and the direct destructive measurements, which is real good news for the validity of photos. We did find this interesting minor overestimation by photos at larger nodes, but we're not too concerned about that because the photos and the direct measurements when ran through our models showed the same optimization. So no matter if the data was from photos or direct, it told the same story about whether or not it was keeping a safety margin, which is critical because at the end of the day, the type of work that we're doing, if it's telling us the same output, whether or not it's keeping a safety margin, that's the bar for whether this data is valid. And thus, photos seemingly can provide valid, useful data. And kind of pulling back a little bit and bringing it back to my kind of journey as an undergraduate, if you will, in my research, I want to talk about some of the opportunities I've had to present it like I am now. First off, I'm currently in the progress of writing a manuscript for a research paper, which I'll be the primary author on, which is, of course, exciting, especially as an undergraduate. I've also had the opportunity to present at multiple conferences, both kind of local and one national conference, Botany. There you can see a nice picture of me presenting at an internal Grand Valley event, and kind of, branching out and talking about what's next. Something I'm excited about, but has been unfortunately kind of put to the side, is some of the tissue work we've been doing. Those are some photos of some tissue samples of leaves and twigs that I took from our plot. That we could pull anatomical data from at some point if we want to. I've also been mining some anatomy data from the literature. Basically, that's cell and microanatomy traits of vasculature and leaf anatomy. And the idea is that someday we might be able to take kind of the microanatomy of leaves and of vascular systems of trees or of individual species and compare those to the different ways that the branching structure is observed in these species. And it's very plausible that these microanatomy things, these physical principles of wood, or just the way the cells in the leaves or vasculature are laid out, might inform how the tree branches at the gross level, which is fascinating. And what I'm working on right now that has been super interesting is looking at this at the gene level. So currently, I'm doing work investigating 2 genes that are known to be involved in the branch angle of tree species. I'm looking at TAC1 and LASY1. And they've been observed in ornamental species and fruit trees to be involved in branch angle, but I'm looking at them in native species. And right now we're dyeing primers, and we're going to go take some samples from the field here soon. But that work will continue into next year. And I do want to shout out Dr. Margaret Dietrich, who is my research mentor for this more molecular side of this. And that's where we're at right now. It's been an exciting journey, and I'm excited to see what I will get into it with the genetic stuff next year. But before I go, I want to say a couple thanks. The biggest one is to Dr. Gary Greer, who has been my research mentor for the past 3 years. He's been a professor of mine. I consider him a friend, and he has taught me so much, and I would not be here without him. I want to thank Dr. Margaret Dietrich, who I just mentioned, who I'm doing my molecular work with on TAC and LASY. I want to thank that whole list of students there who each one of them was involved at some point in the last 3 years with this research. I want to especially shout out Stephen, who did that he cut the tree down for the destructive conformation. I want to thank GVSU University Communications for the nice photos that you saw of me in the plot and at that presentation. They were the ones who took those photos, so I'm super thankful for that. I need to thank Grand Valley State University Office of Undergraduate Research and Scholarship for summer funding for 2020 and for the summer of 2021 with the S-Cubed program. I'm exceptionally grateful for that. I would not have been able to do some manuscript writing or any of that plot work without their funding. And most of all, finally, I want to thank you for watching. I really appreciate you taking the time to hear what's going on here. I hope that you have an excellent rest of Student Scholars Day and an excellent rest of your day. If you have any questions, feel free to reach out with the Student Scholars Day system or just in the YouTube comments. Also, if it's maybe a little bit later, my email is up there on screen. I would really love to hear from you if you've got kind of questions about how I'm doing this or ideas for maybe something else I could look at or a new way of thinking about it. But you have a great rest of your day, and thank you very much. Bye.